Effects of altered IKZF1 expression on CAR T therapy efficacy in mantle cell lymphoma. Mantle cell lymphoma, or MCL, is an aggressive cancer of the lymphatic system that primarily affects B cells, which are responsible for producing antibodies in the immune system. MCL is characterized by abnormal growth of the immature B cells in the lymph nodes, which make up the mantle zone, and accounts for about 6% of all non-Hodgkin's lymphoma cases. One of the most difficult aspects of treating mantle cell lymphoma is its high recurrence rate, meaning that many patients relapse even after initially successful treatment. Current treatment options for mantle cell lymphoma often involved a combination of therapies, including chemotherapy, immunotherapy, stem cell or bone marrow transplants, and CAR T-cell therapy. While chemotherapy can reduce tumor burden, it is highly toxic and damages healthy cells. CAR T therapy has emerged as a more targeted alternative that uses the patient's own immune system to fight cancer. CAR T therapy works by genetically engineering a patient's T cells to express a chimeric antigen receptor, or a CAR, which allows the T cells to recognize a specific antigen on the surface of cancer cells. One major advantage of CAR T therapy is that these engineered T cells can persist in the body for years after initial injection, which provides a long-term prevention against relapse. However, CAR T therapy is not always effective long-term because of a major limitation in CAR T therapy known as antigen escape, where cancer cells reduce or lose expression of the target antigen. In my research, CD19, a protein called CD19, was the target antigen because it is widely expressed on B cells, therefore increasing the chances of the engineered CAR Ts binding to cancerous cells. When cancer cells downregulate CD19, CAR T cells have fewer binding sites, making them less able to recognize and kill the cancer cells. This can ultimately lead to treatment failure and relapse. Because of this, improving or stabilizing CD19 expression on cancer cells could significantly enhance CAR T therapy effectiveness. This led to my focus on IKZF1, a zinc finger transcription factor that binds directly to DNA and plays a critical role in B cell development and differentiation. IKZF1 acts upstream of CD19, meaning it helps to regulate CD19 expression. My research question was whether overexpressing IKZF1 could increase CD19 expression on mantle cell lymphoma cells. I hypothesized that increasing IKZF1 expression would raise CD19 levels on the cell surface, counteracting antigen escape and making cancer cells more susceptible to CAR T cell killing. To test this hypothesis, mantle cell lymphoma cell lines JACO and MINO were expanded in culture media. IKZF1 was overexpressed in these mantle cell lymphoma cells using two independent approaches, a lentiviral transduction method and inhibition of the CK2 signaling pathway, which indirectly increases IKZF1 expression because CK2 is a protein that is upstream of IKZF1, which, when downregulated, leads to its overexpression. Western blot analysis was used to confirm successful overexpression of IKZF1. Next, CAR T cells targeting CD19 were produced from human T cells and co-cultured with either wild-type lymphoma cells or IKZF1 overexpressing cells. Flow cytometry was used to measure surface CD19 expression and to quantify how many cancer cells survived after CAR T treatment. This figure on the left, the results on the left, show that increasing IKZF1 expression led to significantly higher surface CD19 levels. Additionally, this figure on the right shows that the IKZF1 overexpressing cells were more easily killed by CAR T cells, as demonstrated by a lower post-co-culture live cell count. These findings suggest that increasing IKZF1 expression may help overcome antigen escape, a major obstacle in CAR T therapy. Future research will focus on understanding the molecular pathway linking IKZF1 and CD19, as well as testing this approach in animal models with the goal of eventual clinical application. In conclusion, this project demonstrates that targeting gene regulation upstream of CAR T antigens may be a promising strategy to improve immunotherapy outcomes for aggressive cancers such as mantle cell lymphoma. Thank you for your time.